let you know as soon as the recording has started. All right, so the recording has begun. Want to welcome everyone today to LCB's Deliberative Dialogue Session 3 concerning evaluation of THC compounds, and begin a discussion around how we might determine and define what is and is not impairing today. I'll go over the agenda really briefly and then move into our slide deck. I'm just going to have a brief introduction here for the next 5 or 10 minutes or so, going through panelist roles, my role as the moderator, participant and listener roles, and then our meeting format. And then we'll have a brief introduction of our panelists today, and then begin our moderated panel discussion. And then we'll have an open question section starting at 2 p.m. And then we'll wrap up at 2:25 so we can stay on time today. Let me see if I can say that. We'll move right into this. All right, so I'd just like to set the stage for today's meeting. Our goal today, again, is to build an understanding of how we might determine and define what THC compounds are and are not impairing. Our intentions today are the same as many of our deliberative dialogue sessions, but here we'd like to continue a platform to share and discuss plant chemistry perspectives, increase opportunity for respectful and moderated dialogue between all participants, and continue the sessions that we started last summer, so that was last June and July, about these issues to inform future rule and policy development. So why are we here? I'm sure many of you are familiar with why we're here, but we'll go over it again just in case. The agency issued a policy statement last year concerning THC compounds other than Delta-9, and we adopted a new rules section effective February 5th of this year around the evaluation of THC compounds, and we defined terms such as additive, solvent, ingredient, and compound in those rules. 2022 legislative session came and went and didn't result in any changes or any definitions that we could advance in rules. So we'd like to continue a discussion concerning cannabis plant chemistry to inform future rule and policy development. With all of our deliberative dialogue sessions, comments that are received are added to an Excel workbook or a table. They're organized by theme and analyzed. And then we share those with both the public and the board to inform decision-making moving forward. We also, as I noted, record these sessions and put them up on our website so everyone can view them at a time of your convenience or for additional information. All right, so meeting protocol for today. We have 4 fantastic panelists that are rejoining us from our sessions from June and July of last year. I'll be your moderator today. I'm Kathy Hoffman, Policy and Rules Manager for the agency. And then we have all of our great guests who joined us today. You'll be our participants and listeners, and hoping that you can stay in participant and listener mode until 2 o'clock when we start taking questions. And then I would like to just briefly touch on what deliberative dialogue is, although many of you are familiar with us because you've joined us for other deliberative dialogue sessions. But again, just want to really emphasize that this is an opportunity for us to all think together, to listen deeply to other points of view, explore new ideas. We're really looking for points of agreement and examine assumptions that we might have about these particular issues. And then just to emphasize the difference between deliberative dialogue and debate, and I'm going to focus here on the what deliberative dialogue is. Again, it's a way for us to find common ground, to listen and find meaning in the subjects that we're addressing here, consider alternative, and then move the conversation forward. So just again, gonna concentrate on the positives of deliberative dialogue for this session today. We do have a few ground rules. I'm not going to go through all of these because, again, I know many of you have already seen these. But again, I just want to emphasize that there is no right answer here. This is not an opportunity for us to debate each other or seek points of diversion. Really, we're here to find common ground so that we can move the conversation forward around what impairing and non-impairing might mean, if we even use those words at all. Again, this is a public work session. It will be recorded, and we'll put that up on our outward-facing website when we're done today. So a little bit about the format. I'll introduce the panel here in a few moments, and each panelist will introduce themselves, give a little bit of background, and then we'll begin the moderated panel discussion. So that's where participants listen to the panel discussing issues amongst themselves, and then I'll try to ask a few questions, and we're hoping for a really good dialogue around this topic today. And then once that discussion is over, and again, we hope that that will be concluded around 2 p.m., then we'll have a question and answer or discussion session. And so if you would just kindly raise your hand, use the hand raise function in Teams, and then either Jeff or Audrey will call on you in the order that your hand was received, or it was held up, I guess is the best way to say it. I just want to take a brief moment here and introduce who from LCB is helping to make this session happen today. We've got our rules coordinators, Jeff Kildall and Audrey Basic, joining today, our administrative professional, Tierney Hamilton Steele, and our policy and rules director, Justin Nordhorn. And I just wanted to stop here for a moment and ask Justin if there's anything that you wanted to add or say before we get started today.
Thanks, Kathy. I think you covered everything really well. The only thing I would add is please keep in mind, we're planning on a number of these deliberative dialogues. So this is not going to be the only session for feedback and on topics. So we are planning additional deliberative dialogues in the near future. So not everything is going to be covered today. And so if you have interest in those, please watch out for those particular topics. So thank you.
Fantastic. Thanks, Justin. And when we conclude today, I do have a slide with the tentative dates for our future deliberative dialogues. There's one scheduled in May and June. All right, so I'm going to do two things at once here. If you feel compelled, please share who you are. If you're affiliated with any association, if you wish to share that in the chat box and maybe give a little brief sentence or two about why you're interested in this topic. And then I'd like to call our great panelists to come on camera. And we'll go ahead and start with introductions. And Neffy, since you are already on camera, we'll start with you.
Okay, thank you. So yes, I'm Neffy Stella. I'm a professor in pharmacology and psychiatry and behavioral sciences at the University of Washington.
Great, thank you, Neffy. And Jessica.
My name is Jessica Tanani. I'm CEO of VertiBio Research. We have a research license in the state of Washington, and we focus mostly on molecular biology of the plant and metabolite analysis.
Fantastic. Thank you, Jessica. And Brad.
Hi, everyone. Brad Douglas, organic medicinal chemist by training, and professionally I've been involved with regulatory affairs, namely FDA affairs, for the last 15, 20 years. Great to be here.
Thanks, Brad. And then David.
Hi, my name is David Gang. I'm a professor at Washington State University in the Institute of Biological Chemistry. I'm also the director of WSU's Center for Cannabis Policy Research and Outreach.
Thank you. Thank you, panelists, for joining us today. And if you weren't here for our previous deliberative dialogue sessions, this was our original panel. So we're just really glad that you're able to join us to kick us off on this sort of new discussion we're having around what is and is not impairing. So we're right on schedule. I'm really happy about that. I'd like to go ahead and kick us off with our overarching question, and that is, how might we determine and define what is and is not impairing? And I think when we've thought about this concept, certainly it was something that came up during the legislative session. Things that I think about when considering what is and is not impairing. What is it about the interaction or the brain's interaction with cannabinoids that we're trying to get at when we're thinking about what is and is not impairing? So maybe that is a starting point. And I'm going to turn off the screen so the focus is on our discussion and not the screen.
I think I can kind of kick off things a little bit and then kick it over to other people. I think that one of the biggest things that we've had as a panelist in our discussions is defining the word impairing, whether we use the word impairing or intoxicating or psychoactive, or there's not really a good word to define what we all kind of know as the unknown. So, there's an interaction that happens in the brain that makes people are the probability that they could be high. And that probability that they could be high, I think, is what we're defining. And so the word impairing, I think people have, potentially an immediate negative response to the word impairing, as it may not be perfect. But I think, I would like to propose for the discussion today that we use the word impairing, even though we know it's not perfect, with that caveat. I don't know if anybody else has any other comments on that to start.
Sounds good. I'm in agreement. I would love to hear from Neffy, being the pharmacologist of our bunch here, regarding what is it on a receptor level or a pharmacological pathway level. What is happening when THC or a similar molecule is making somebody feel high, is causing impairment?
Okay, I'll try to decorticate this. So I'll agree with using the definition of impairing. I think it's a good one just to kind of set the stage. And it's a good way to actually start with the pharmacology. So when individuals take cannabis-based products, and we'll be focusing mainly on the impairing properties of THC, because CBD has a completely different bioactivity. THC will eventually reach the brain and interact with the cannabinoid receptors that are expressed in multiple areas of the brain. And I think the fact that it's in multiple areas of the brain is very important, because the way people talk about the activity of THC on the brain is what we say polymodal. It does many different things. And therefore, right off the bat, I think we should differentiate the euphoric effect, which is what people actually refer to getting high, to the impairing effect. And the impairing effect is going to be hard to define, because it'll depend on the type of task that the individual are performing. So in the cannabinoid field, when we start talking about impairments, we start thinking about motor impairments. So this is already the connection with driving. Are we going to have some motor impairments? Are we going to have, is our reaction times going to be different? Is the decision making going to be affected? Or is our ability to focus going to be affected? So those are all the type of questions that people think about when we use the word impairing. And in terms of the pharmacology, it's because we have these receptors in the different areas of the brain, and they are activated by THC, and therefore that's why this area of the brain is affected, and therefore you might have euphoria from one side, motor impairment, lack of focus, and all these different effects. So I think impairing is a multiple. There's multiple aspects in impairing, and it'll depend on the type of task that the individual is performing, if they are truly impaired or not. So that's my first real quick answer.
And it's my understanding, Neffy, that the CB1 receptor mediates the euphoric effects. Does it
Correct.
also generally mediate the impairing effects, or is
Absolutely. Absolutely. So the THC is going to interact with the cannabinoid CB1 receptors on neurons that control attention in one part of the brain, on neurons that are in the cortex, and that's the sensory awareness. Music is enhanced, colors is enhanced, sounds are enhanced. That's the euphoric effect, enhanced sensory awareness. And then there's other receptors in the more deeper parts of the brain, in what we call the basal ganglia, that are involved in other types of function, which is motor coordinations, motor precision. So the fact that these receptors in all these areas of the brain are interacted by THC will affect all these different behaviors, but it's all through CB1 receptor. The difference is that CB1 receptors is expressed in distinct part of the brain.
And I think one of the things that we struggled with a little bit last legislative session is kind of defining the fact that it's not just Delta 9, that there's Delta 8, Delta 10, a wide variety of these. Neffy, can you address at all the fact that some of these other compounds still bind the CB1 receptor at decent affinities and can cause impairment, for lack of a better word?
Yeah, absolutely. So yes, so that would be the primary compound. It would be Delta 9, Delta 8, and Delta 10. They all have similar activity or comparable activity at the CB1 receptor, according to the results that we have at this day. The field has just started to actually pay attention and start to study Delta 8 and Delta 10. So we're gathering new results on how comparable is the activity of Delta 8 and Delta 10 to Delta 9. So the current understanding is based on older literature, but it's being confirmed today. So these three initial compounds will have comparable activities on the CB1 receptor. I think another way, one recent hypothesis that has risen in the field is that, when individuals use THC, then it actually is going to be metabolized by the liver. And the first metabolite of THC is going to be 11-hydroxy THC. And that's actually used for pharmacokinetic drug testing tests in urine and things like that to understand the metabolism. Well, the field is actually starting to realize that because 11-hydroxy THC is also active at the CB1 receptor, that when individuals are actually taking THC, the effect of THC is not stopped by its metabolism, because the first metabolite, 11-hydroxy THC, is still active at the CB1 receptor. And that actually might be one of the reasons why, even though when THC goes up and down in the body, individuals are still feeling the psychotropic effect. It's because there's a lingering of the 11-hydroxy that is still activating the CB1 receptor. And that same hypothesis of Delta 9 being metabolized, the 11-hydroxy, we actually have no idea what is the metabolism of Delta 8, what is the metabolism of Delta 10, and those metabolites might also have some bioactivity. But I don't, so in terms of, go ahead.
Just quick follow-up question. Does the mode of ingestion change the metabolism at all, Neffy?
The speed at which it's metabolized, yes. So as individuals are using it through the lungs, inhaled, either smoking or vaping, then it goes directly into the lungs and then directly through the heart and to the brain. So there's no I just muted, sorry. Let me repeat. So there's as the molecule goes in, as the THC goes through the lungs, it goes directly into the blood, through the heart, and then directly through the brain. So this is why the effect of inhaled THC is very rapid. The psychotropic effect is very rapid. When it's taken, and then eventually, as it comes out of the brain, it goes back into circulation, and then it goes through the liver and starts to be metabolized. So when it's inhaled, there is no first pass metabolism. It goes directly to the brain. Whereas when it's taken orally, then it goes into the gut. It's actually digested. We probably lose a lot of the compound with the acidity of the pancreas. And then it goes into our bloodstream through the liver, and it has the first pass metabolism. So actually, the THC that is absorbed through the oral route, there's only a fraction of it that is going to reach the brain because it's already metabolized by the liver. That first metabolite is 11 hydroxy THC, and that might also be actually be producing some of the psychotropic effect.
You know, I've heard, Effie, in the literature, there's been estimates that the 11 hydroxy THC can be, you know, significantly more psychotropic, impairing perhaps than delta 9 THC itself. Do we know receptor binding affinity for the CB1 receptor versus delta 9 THC?
Absolutely, yeah, we do, and actually this has been known since like the 90s, where we know that delta, the 11 hydroxy, or even the delta 8, can bind to the receptor. But in pharmacology, there's two components that we need to pay attention to. The first component is the binding of the molecule to the receptor. And that's the affinity, and that's where we start talking about this is a very potent compound and things, because very low concentration of compounds can activate the receptor. So that's the first part, and we understand that pretty well since the 90s. However, now we actually are understanding that these cannabinoid receptor, once they're activated by different compounds, they can do slightly different things on the neurons, and therefore at this day, we actually have very little understanding besides the fact that 11 hydroxy or delta 8 or delta 10 can bind to the receptor. We actually do not really understand how well it activates the receptor. And that could be linked to what you're actually suggesting, that maybe 11 hydroxy could be more potent, meaning it could bind the same way as delta 9 THC on the receptor, but the effect later on the receptor might actually be more. So those are all, I'm pushing the science a little bit too far right now, because I'm basing this, what I'm saying right now, on what we understand on the binding that is 20 years old, and just on the recent novel experiments. So I think there's a tiny bit, there's probably a little bit of more research to be done to actually solidify this. But yeah, so those are the two things that we have to consider. One is, where are the receptors expressed in the brain in terms of impairment, meaning it'll change this brain function versus this brain function. Then the second thing we need to consider is the molecule that is going to reach the receptor, delta 9, 11 hydroxy, delta 8, and delta 10. How precisely is that receptor going to be activated? Is delta 9 going to be a weaker agonist than 11 hydroxy? We actually don't know that. We're just starting to figure that out.
And I think one of the discussion points we had kind of in the prep is, you know, I think as a society we're so used to alcohol, which is kind of a direct correlation, and this is a little bit more, you know, a little bit more difficult to make that direct correlation from level to impairment. And that's why I think there's been such an active debate about how to define the molecules. I don't know if anybody else, you know, David, have comments on that, but it seems difficult in general.
Yeah, I would agree with that. And I was going to ask David if you had anything you wanted to add so far.
Oh, I've been happy listening to Neffy talk. It's great.
Sure.
No, but yeah, actually something that I thought of, that Jessica, your question is kind of leads into exactly what I was going to say. And that is that when you think about what Neffy just said, you know, this is an important thing to point out too to everybody that's listening. Delta 9, there's one version of it, the negative stereoisomer of it, right? The negative optical rotation, the S stereoisomer of it, that's the active. That's the version that's active. Same goes for the delta 8 and the delta 10. So there's a single molecule, a single version of these molecules. And we talked about this in the previous deliberative dialogue, right? The versions we're talking about, it's kind of like our left and right hands, right? So the S version is like your left-handed version. That's the one that's active. And it's binding to like the receptor is kind of like a glove that fits over your hand. Okay, and it has to fit just right for it to get in there. If the receptor only has a place for 4 fingers and you got 5 fingers, your hand doesn't go in very well, right? Or if you have a big floppy mitten that you put your hand into, it can go in, but it doesn't stick very well. And that's kind of like how the receptor binding might be viewed, as maybe an okay analogy there. But what's the important thing is that molecule that goes in, that hand that goes in, it's identical. It's the same exact chemical entity every single time. But what's different is the glove. And each individual that's going to take this compound, one of these compounds, by smoking it or ingesting it, swallowing it, whatever, their bodies are all different from each other. We have different gut microflora that are going to metabolize and work on those compounds differently. Our livers are different. We have different enzymes. We know what some of the enzymes are now that are involved in that detoxification of the compound. It's a detoxification pathway that our liver does that converts these foreign molecules into a form that can be eliminated from the body. That's how it gets eliminated out. And each person has slightly different versions of those enzymes, right? Because we all are genetically not identical. So the enzymes are proteins and they're made out of DNA. They're coded by DNA made out of amino acid sequences, and those vary a little bit from individual to individual. And those slight changes in the amino acid sequence of those proteins changes the structure just a little bit. Sometimes it can change it more than just a little bit. So different individuals are going to respond. Their enzyme, their receptor, the enzyme in the liver that changes it, or the receptor, could potentially be a little bit different from somebody else. So that means that, and then those receptors are on their neurons. Those neurons were developed differently because we all have different brains. All of those factors come into play when you take one of these. And so each person can have a different response. Now, it's in general very similar, right? It's not completely different, but it's different enough that that's one of the reasons why we're having this conversation. It's different enough that it's hard to define where it is that somebody gets affected enough that everybody else in society starts to get worried about it. I mean, that's what we're really talking about here, right? It's like drunk driving, any kind of impairment, or under the influence, whatever words you want to use there, is we get worried about somebody else taking this, doing something that could then impact somebody else, like me or you or our children or whatever, right? That's why we get concerned about this. That's why we're having this conversation. And because everybody's a little bit different, we're all going to respond a little bit differently. And these molecules also, they hang around in the body differently than alcohol does. Alcohol is a very, very simple molecule, right? Very, very, very simple. The cannabinoids are not. They're more complex. So their metabolism's more complicated. It's more difficult. The timing, it takes longer. All of those things come into play. And that affects how somebody is going to respond to this. That affects, and also we know that the body gets changed.
Actually, wait for me. Give me 1 minute.
Somebody's talking. Okay. The body your body gets changed over time when you're exposed to something like this. So, it's very well known that somebody that's a long-term consumer of these compounds has a different response than somebody who's never taken it before and it's their first exposure. And that's because their liver's different, their brain's different, and their exposure and their sensitization to it or desensitization to them. All of those factors come into play. And it makes it much more difficult to try to figure out, okay, when does somebody become affected enough that we need to worry about it? But we obviously do, because everybody thinks these need to be regulated. But the challenge is, where do you draw that line? How do you figure out what that line is? When do we know that this is an issue?
And what I think I'm hearing you say is that different enough can be different for every person. That's what I think I just heard you say.
The data seems to show that. There have been studies done with people looking at trying to figure out how you measure this. Like Neffy mentioned, we've looked at urine samples, we've looked at blood samples, and if you try to look at doses that you give to different people versus those results, there's not a very strong correlation. With alcohol, there's a nice correlation between how many beers you drink, how many ounces of scotch you drink, whatever, versus the blood alcohol level. There's a pretty good correlation there. But with the cannabinoids and THC, that doesn't seem to be the case.
And I think, the extra thing in this is the fact that not all cannabinoids do this. So it's,
Oh, well, that's
it's,
another
you
thing
know,
we're
that,
gonna get, yeah.
so it's one of those things that we have to figure out how to, define the cannabinoids that do these effects versus the cannabinoids that don't, and what level should they be, deemed impairing, I guess. So it's almost two things of, and I think that's why we've led to some degree of a structure. It has to have a structure of a cannabinoid, but it also has to have the function of binding to the receptor. I don't know if.
Yeah, because it seems that that combination is really important for it to have the activity we're talking about. Just because we put the word cannabinol as a cannabinoid as a classification doesn't mean they have the same function. Something that's got the word alcohol, for example, in it doesn't mean that it's the same thing, right? So alcohol that you drink versus a sugar alcohol like sorbitol, it's chemically defined as an alcohol, but you add sorbitol to, lollipops, and it's a solid, and it has no impact on your brain or anything, right? It's complete, but it's an alcohol, but it has no impact on impairment. It doesn't do any of those kinds of things at all. But chemically defined, you can call it an alcohol, right? There's a whole bunch of those we come up with. But yeah, it's sometimes terms that we use to classify things can also kind of muddy the waters a little bit.
Maybe I'll build a little bit on what was said. I think probably the impairing effect should not be focused on the compounds, but rather on what activates the CB1 receptor, because this is what actually is, because you have some artificial cannabinoids that are in spice compounds, spice products that don't have the same chemical structure, and they're very potent at the CB1 receptor and therefore will be very impairing. So truly impairing will be associated with activation of the CB1 receptor. To build on what David was saying about the diversity in the individuals, not only there's a genetic difference, but there's really a history difference. The liver is, as David was saying, is the main goal is to detoxify foreign compounds. The more you're exposed to these molecules, the more liver will actually upregulate these enzymes to degrade them. So a person who's actually been exposed to THC for a long amount of time will have actually a very active liver that metabolizes because it's been exposed, it's been upregulated. So that might be another difference between individuals. The amount of THC that this person has taken will change the liver metabolism. Or even somebody who actually has a liver disease will metabolize. So then there's the health aspect of the individual that comes in. So that's in terms of the liver. There's exactly the same phenomenon at the level of the brain, at the level of the receptor, which is called tolerance. The first time individuals use THC, all the receptors are there ready to go and they get activated, and the psychotropic effect is going to be pretty strong. However, the more the individuals are actually exposed to the cannabinoid compounds and activate the cannabinoid receptors, these receptors will undergo tolerance, meaning they're not going to be working anymore. The image might be removed from the neurons. So that's why actually there's this phenomenon of individuals who get high the first time when they start, and then it doesn't work as well, and they need higher concentration, and they escalate in the amount of THC. So the level of impairment, this is what people call the functional impaired person, right? This person who's been using THC for so long that they're so tolerant that they can use very high doses of THC even before getting impaired. So I think the diversity in how individuals are going to be responding is very, very is going to be very vast. The other thing I want to emphasize is the link between alcohol, in line with what Jessica actually was saying. There is, and David, there's a well-established relationship between the amount of alcohol in the blood and the type of impairment that it does. We'll do the little test and things like that. Because alcohol has a very different impairing activity than THC, the typical stories we hear are people that are drunk are very aggressive and will drive very fast, whereas people that are high are very paranoid and will drive very slowly. Very rarely are firemen have these stories that very rarely a fire is started by a person who was high because they're very relaxed. It's mainly an accident because people were drunk. So the behavioral impairments between alcohol and THC are very, very different. And this is where the field is going. This is what we're doing in our lab. Actually, we're trying to understand the impairing properties of THC on our little mice. How do they, are their movement impaired? Is their focus impaired? All these things so that we can maybe translate that and have a better understanding of what's happening in humans. But we already know that the impairing effect of THC and alcohol are completely different. The type of question, the papers that we're currently reading on THC-induced impairment in humans, in 20 years ago, the questionnaire is, how do you feel? Do you feel high right now? A little bit, medium, high. So very basic description of the psychotropic effect. Now the field understands that there's a need to better understand the impairing effect in humans, and therefore we're creating new questionnaires that are more tailored to the THC impairing properties, and even devices to be able to actually correlate how much THC corresponds to this impairment so that we can try to reproduce something similar to what the alcohol field has done with the testing. And then finally, the fact that these cannabinoids are actually hydrophobic, so very greasy molecules compared to alcohol, which is volatile. So alcohol makes sense. It's volatile. It'll be excreted by the lungs, and we can have a breathalyzer. And we can measure precisely how much alcohol corresponds to the blood. But because THC and cannabinoids are lipophilic, greasy molecules, they're not evacuated by the lungs, and therefore the idea of developing a breathalyzer for THC is going to be very, very challenging. Probably the route to go is going to be to actually measure the amount of THC or their metabolites in blood in a very acute session, and then we might actually start getting a better correlation between the amount of THC in blood and the behavioral impairment. Lots of concepts.
Can I ask you a little bit more about that, Neffy? With the advances now in functional impairment and tying that to THC levels or metabolites in the blood, do you think that the animal models that are currently available to assess the impairment or the activity at CB1 are still useful as a proxy for assessing impairment? Or have there been enough advances in humans and the work that you're doing and others that the things like the tetrad test are not necessarily as useful?
Yeah, that's a good question. So the field is now working in parallel with the animal models and the humans, and we're communicating to try to identify what is impairment in humans and in animals. So there are things that are easy to test in a mouse, which is motor impairment, focus, all these really basic functions. But decision making, time perception, all these other things that are really important in humans are much more challenging to test in rodents. So we can help until a certain limit, a certain amount of behaviors, but then the final stretch will have to be done in humans. There's some really interesting work that is coming out now in testing the bioactivity of THC in humans, where they're doing actually functional imaging of the brain. Try to even understand which part of the brain is actually affected when THC is used. And there's a really big focus currently on the prefrontal cortex, which is truly the decision making. Chess players have a fantastic prefrontal cortex because they can strategize and things like that. So, but if you're under the influence and if you've taken THC and you're impaired in your prefrontal cortex, that'll be a decision-making impairment. I don't think that's much harder to actually test in mice. So the good news is the field is actually working hard in parallel. I'm seeing the papers come out, and there's some good communication here.
Is it fair to say, I know we had this discussion sort of in the back room, and we are in a state that has a per C blood level for THC. Is it fair to say that there is not good correlation between blood levels, breath levels, you mentioned Neffy, urine levels, whatever the case may be, and any sort of functional impairment in humans? Is that the state of the art currently?
Yeah, we don't have a definite answer right now. People are working towards, we have initial ideas of in what direction it's going to be, but I agree. The current correlation between the drug levels in blood and the human impairment are not actually validated. There's still quite a bit of validation. And if we're thinking about impairment in terms of driving, which is a simple way of thinking, there's some research now where individuals are actually administering THC to individuals, and then they do virtual driving, and they play these video games, and then you kind of see how they drive really slow, for example. And they're trying to understand what is the type of impairment that you can get in a video game, and that will definitely be translated into the route. But again, it's about what is the task at stake here. And being a pharmacologist, it's always about the dose. How much has actually been taken, and what's the prior history? Some people might take a couple puffs on a joint and be completely impaired, and another individual who has much more experience and is much tolerant can smoke out a whole joint and not be as impaired. So that's another aspect that needs to be considered.
Well, I'd like to stop here just briefly and reach out to Justin, because I know you're probably taking notes along with me. Justin, are there any questions that you'd like to pose to the panel that have come up for you?
Sure. When we're discussing the impairment issue, I think one of the important dynamics that we get questions on, especially from legislators and others, is how do you tell the difference, not just by the chemical compound, but and I agree with the stuff I've listened to and read. I can understand where you're coming from when you're talking about the CB1 receptor activity. But how do you differentiate an impairing product versus a non-impairing product at a threshold? And as the example, when we think about alcohol, anything that is above 0.5% alcohol by volume is considered to be an alcoholic beverage, therefore regulated and age restricted. So that's a really low threshold to be alcohol. So what would a threshold look like in a cannabis type of product? Because we know that there's entourage effects when you're introducing two cannabinoids together. One can have an effect on the other. So what's your thoughts around how do you make a distinction between an impairing product and others?
Justin, this is Jessica. I think one of the first important things is defining what an impairing cannabinoid is. And then you can look at what should a serving size limit be, packaging, that kind of thing. But I think that, one of the biggest things I think we need to do is how do we define what an impairing cannabinoid is? And there's a decent list of them, I think, today, but people are pretty creative with chemistry. So if we can come up with a definition of a chemical structure and a binding to the CB1 receptor, then we can look at how much of that leads to impairment. But I think to a large degree right now, we don't even have, I guess we have the CSA in the state of Washington that lists a number of these, but we really don't, from an LCB perspective, have that list that I know of. I don't know if anybody else on the panel has feedback on that.
Not really, and I'd say that nobody really does, but I will introduce a concept that we think about from a medical chemistry standpoint, and that's structure-activity relationships. We know with whatever pharmacological compound or class of compounds that are active somewhere in the body, that changing small parts of these class of molecules, either enhances effects or can decrease effects. So with THC-like molecules, you know there's a couple places on these molecules that if you change, you tweak them, that you're going to still have the impairing CB1 agonist activity. And I think if we're putting a list together of compounds that may be impairing, we have to focus on these particular pharmacophores, these particular areas of these THC-like compounds that are known to retain high inducing or impairing activity.
Yeah, for example, we mentioned earlier the 11-hydroxy form, right? So if you put a hydroxyl group on that position, you don't lose activity. Maybe it actually enhances it in some cases, in some individuals. But if you take a molecule that's similar in structure, for example, CBD, that's one everybody talks about all the time, and you try to show that it has the same activity as the THCs, and you find it doesn't, right? You don't get high. No matter how much you give somebody, they don't get high. It doesn't impair the driving the same way. It doesn't impair the decision-making process the same way. It doesn't do any of the same things that THC does. So, and most of the structure is very similar, if not identical, to THC, but it's got a few changes that are pretty significant that make it a different subclass, and therefore it has a very different activity. And there's a whole bunch of, most of the, I think we could say the vast majority of the other cannabinoids fit that definition too. They don't have the same impairing effect, if we're going to use the word impairing, whatever that means. They don't do the same thing to the body, to the brain, and they don't function the same way. But they might make you a little bit more relaxed, or if given at a high enough dose, they might make you a little bit sleepy. But we had a conversation about this in a previous deliberative dialogue. You can come up with a list of any number of compounds, food additives, whatever, that if you take enough of, they do the same exact thing, and none of those are regulated. So, right, so where do you draw the line about what is that? We're back to that point, like Justin was saying, the point about with alcohol, there's a level, and that's because we want to make sure we can regulate it, because we know that once you exceed that threshold, you get to a certain point, it starts to become a problem. And if somebody drinks enough alcohol at low enough dose, at low enough concentration, then, especially if they haven't drank it much before, then they could potentially be affected. But if you switch categories, you switch it to a different molecule that they eat or drink or whatever, you're not going to have the same effect. So,
and I think that's important too, David, here, back to your question, Justin, that with ethanol, alcohol, we're dealing with one molecule. So it's easier to set limits or concentration limits on what's an alcoholic beverage or not. With cannabinoids, although some of these THC molecules are very similar molecularly, they can have different impairing effects and vastly depending on the dose. And I'll give you an example. Delta-9 THC has a 5-carbon alkyl chain off of the resorcinol molecule, or the moiety. You add 2 carbons, 2 methylene groups to that, you get a molecule called tetrahydrocannabiphorol. This was recently discovered to be found naturally in cannabis a few years ago. That can have a significantly greater impairing effect, significantly greater agonist activity at CB1 receptors. So it's very important, I think, that we don't seek out a one-size-fits-all dose ranging, and impairing definition for all of these compounds. They have to be addressed individually.
which makes it challenging.
Yeah, I can see from some perspectives, I guess, from a lay policy kind of perspective that we need to be able to tackle is how do business owners, how does the general public, when they look at a package, they know that yes, this is, or no, this isn't likely to cause impairment for me. And we know that there's, go back to your CBD comment. We know a combination, or you believe the combination of CBD to THC in the right ratio is going to diminish the impairing effect, whereas that same amount of THC may be impairing. And so when you're looking at those things, we have to be able to say, well, it can't just be a molecular compound. Nobody's going to know how to regulate that and see that in a store. How is a reasonable person supposed to understand that? And so I guess that's kind of the threshold question that we're looking at on, it's not one size fits all. I totally agree with you on that, but I'm trying to figure out how do we get to a commonality in that range. And so that's really the struggling question that we have around these, because we're not interested in, and nor is anybody else interested in having organizations or regulators regulate non-impairing products. That's food or whatever. That's somebody else's thing. And what we're trying to respond to is those public health and safety issues. And so when does it become an issue? And that's kind of that threshold that we're looking at.
So let me go for it,
Sorry.
Jessica.
I'll say quickly, I think that if we can define the affinity which we think below which people will get high on a receptor level, that we can then package, figure out what the labeling is on packaging. I think that from a consumer safety perspective, right now we haven't even defined what those impairing cannabinoids are. And if we can, from today, working forward in the future, define what the impairing cannabinoids are, then that becomes how do you inform consumers on the packaging perspective, or what are the thresholds? But I think we don't even have that. What are those molecules yet? And then I'll let Neffy go. Sorry.
No, that's okay. It was exactly in line with what I was thinking. What we talked about right now is only one part of the equation, which is the molecule acting on the receptor. What we don't know is what impairment moves. And if I understand well, in the alcohol field, we have a very good understanding of this level of alcohol in the blood will create this impairment. I cannot touch my nose anymore. I cannot walk on the line anymore. And if I remember well, the alcohol drug testing by the road is two items. One is the breathalyzer so I can get a level. And then the second thing is, are you truly impaired? Can you still aim for your nose? The thing is that when you're impaired with cannabis, depending on the individual, touching your nose is not going to be the behavioral impairment that we're looking for. We don't know what it is. So until we're able to actually establish this level of THC, just if we focused on one molecule, delta-9 THC, this level of delta-9 THC in the blood corresponds to this specific impairment, which is dangerous for driving. Until we have that, it'll be actually hard to establish that limit on the package because we don't know what it is. And it's going to be challenging. That's fine. We can just work really hard. But it's going to be challenging because of the diversity in response between individual. If we had to look in our crystal ball and predict the future, I think that the diversity in behavioral impairment that we're going to get with THC is going to be broader than the behavioral impairment that you get with alcohol, because alcohol is a very simple molecule. THC is a very complicated set of molecule that varies quite a bit in its bioactivity in individuals. For example, some individual gets anxiety and they're going to be scared when they're under using THC, and other people's not at all. So very different types of responses between the individuals.
So last question, Kathy, on the molecular structure. So one of the things that we see that's recognized as impairing from a number of articles and whatnot and industry is, something similar to hexahydrocannabinol. So it's supposed to have that same type of molecular structure as tetrahydrocannabinol without the double bonds. And so when these types of cannabinoids get discovered, how do you work those into this equation on being able to identify that? And of course, obviously, you're going to go back to the CB1 agonist activity, but how do we know those as these new evolving cannabinoids are discovered? How does that fit in the picture? So, and I'll stop there, Kathy.
Well, maybe we could describe how the DEA puts molecule on the Schedule 1 license. The way you do that is you seize a new molecule and you don't know if it's a cannabinoid agonist. So the first thing probably is the chemist, they try to figure out the chemical structure. The second thing they do is does it bind and activate the CB1 receptor? That's the second. So we know it's a cannabinoid CB1 agonist. And then the third thing they do is they ask our little mice, because our mice are really good at being able to discriminate between having access to THC versus not THC. So there's these mice and Research Triangle on the East Coast that every day self-administer a little bit of THC by pressing this lever. And this lever, we know it's not THC. And the next day, it's the other way around. So they're able to know which lever is THC. And then suddenly the DEA arrives with a new molecule, puts it into the experiment, and it's the mouse that is going to say, does this molecule feel like THC or not? That's the level of psychoactivity. And it's not impairment. It just feels like THC for a mouse. And that's sufficient for the drug to go into the Schedule 1 license. I don't argue that this is the right way to go. I think we actually really need to improve this. But the process, figuring out the chemical structure so that we know what we're talking about. Second step, does it activate the CB1 receptor? Yes. Third step, does it create the bioactivity, the psychotropic effects that we're expecting from THC? Is it more? Then let's regulate it such. Is it less? Then let's regulate it such, would be one way to think about it. So we need a behavioral input output, a behavioral readout beyond the CB1 receptor.
Yeah, and one thing to add to that is that, to kind of reiterate something said earlier too, is that each one of these molecules is a different chemical entity. And Justin, you're absolutely right. There's going to be different interactions between those specific compounds and other compounds. Like you said, the THC versus CBD ratio, that has an impact. I mean, we see that with alcohol and other pharmaceutical drugs that people sometimes take, prescriptions that they get. That there's warning labels. Don't take this if you're taking alcohol, or don't drive, or other things. There's lots. We know about drug interactions all the time. There's lots of those that have been documented. It's something that we always have to be worried about. But in the case of the cannabinoids, and as new ones, like you mentioned, the hexahydrocannabinol that are identified, we kind of have to treat each of those as a new molecule. They're not exactly the same thing as something else that we've seen before. It'd be nice if we could. It'd be nice if we could say, okay, it looks sort of kind of like a bird, therefore all birds we're going to regulate the same. Well, but they're not all the same, right? You got chickens, you got ducks, you got storks, you got whatever, penguins. They all do different things. And the cannabinoids are kind of like that too. Some of them are going to be the problem, some of them aren't. But you don't know until you do the things that Neffy just described. And so even though that's a challenge, I think, and it's going to take effort and it's going to be costly, probably somebody's got to do the work for that to figure that out. But we do need to figure out which ones actually have these functions. And you can't just treat every single one because it fits into a general chemical class as the same. I know we're already talking on the same language here. I just want to make sure that we're all on the same page with that. But it does make it difficult. And I wish, and I've thought about this a lot, I wish there was a way to think of an easy way to define these things so that we could just say, okay, here's what we do, here's how we regulate them. But I think I think we could say, well, once we can put it into the bin of being something we want to regulate, then it's pretty straightforward how we regulate it. Right? The question is, does it fit in that bin or does it fit in a different bucket? How do we know where it goes? I think that's the challenge.
And that's where I would say that the functional test that Neffy's describing, not just receptor binding, the functional test allows whatever policy framework for judging and impairing, allows it to be future proof. So instead of doing something like Schedule 1 for DEA, or even using the Analog Act, which is strictly structural, if you have a good definition of impairing of a functional test, something new comes along that fits in that structural class, then you can evaluate, is HHC impairing? Well, if we have a very well-defined functional impairment classification, then it could be immediately in this bucket or not in this bucket. If you don't have that functional test, you're kind of stuck like the DEA has found, or federal policymakers. They play a game of cat and mouse. You say, okay, this molecule's new, we have to address it and go through multiple years of sort of policy to kind of add it to a list. And that just doesn't create clarity for anybody.
And
well,
I think
solve the last.
Oh, sorry. I was going to say the other thing I think is it's important to, as Neffy brought up, I think earlier, artificials are in kind of their own little bucket. And so, this what we're attempting to do right now is about cannabinoids that naturally occur. Anything that doesn't naturally occur is kind of in its own little bucket off to the side. And I think that that's, super important for people to understand and acknowledge, is that people can create artificial cannabinoids, but that, any definition that's been put forward so far has had artificial cannabinoids in their own little bucket, not allowed. And so, we are really only talking about natural occurring cannabinoids, and there's still a large class of those, but I think that that's important to articulate.
So I want to move into the question section from our audience. We've run a little over. I kind of suspected that would happen, but that's all right. It's been an excellent conversation, and let's see if we can make it better by some additional questions from our participants. So calling on Jeff and Audrey, I think perhaps the two of you have been monitoring the chat box, and then we'll also entertain hands being raised. So inviting Jeff and Audrey to let us know what's been going on in that chat box and whether or not hands are raised.
Hi, Kathy. Yeah, this is Audrey. There's no
Hi.
hands at the moment.
Okay.
We did receive 2 questions in the chat. The first question was from Gabriel Picard, asking where we could, if we could share the links for past deliberative dialogue recordings. So we'll have to follow up on that.
Okay.
And I don't think Gabriel's currently on, but if you are and you had more to add, please go ahead and raise your hand. The second question we got was from Sean Dene, and Sean Dene, if you wanted to just speak to your question, please feel free to unmute and go ahead. Okay, I'll just read it out loud. And Sean, if you want to raise your hand and follow up, please do so. So Sean wrote, is there research on how minor cannabinoids, in parentheses, CBG, CBN, etc., consumed in isolates at significant levels, greater than 10%, affect the endocannabinoid system?
That's a very exciting question, actually. The field is really starting to pay attention to that. The short answer is probably, well, CBN is thought to actually interact with the cannabinoid CB2 receptor. So CBN might actually have more therapeutic anti-inflammatory properties. CBN does not interact with the CB1 receptor, and therefore it's not going to be producing any of the psychotropic effects. CBG is the new fascinating compound in the cannabinoid field. We know that it has some interesting, very different bioactivity profile. So it does different things. So we know that it doesn't interact with the CB1 receptor. It does not interact with the CB2 receptor on immune cells. So now the field is starting to ask, so what is the receptor that it's interacting with, and what are the biological functions that it's going to regulate? So as the field has grown, we focused so much attention on THC, and then CBD showed up and we started to understand. I predict that CBG is going to be the next cannabinoid that people are going to start to study, but it doesn't appear to have any impairing effects that are similar to THC because it has a different molecular mechanism of action.
Kathy, just to follow up on that, nothing conclusive yet, though. Still, I think I heard you say it may.
Well, we know what it doesn't do. We know that CBG does not interact with the CB1 receptor, and therefore it's not going to be affecting the endocannabinoid signaling pathway, because the endogenous cannabinoid signaling pathway works through CB1 and CB2. So because CBG is acting through a different receptor, we don't know the receptor, and we don't know what endogenous pathway is affected by that.
Okay, thank you.
You're welcome.
So one clarification on that, Neffy. Knowing that CBG and CBN are not agonists at the CB1 receptor, is it fair to say that we could classify both of those individual compounds as not impairing in a THC-like way?
Correct, yeah. That would be my conclusion, absolutely. The same as CBD. CBD actually is not an agonist at the CB1 receptor. Actually, it is called, there's two hypotheses of how CBD can moderate the effect of THC on the CB1 receptor. The first hypothesis is that CBD binds to another binding site, which is called the negative allosteric binding site. And therefore, that's what it does. It tunes down the activity of CB1 receptor, even though it's activated with THC. So that's the first hypothesis. The second hypothesis is CBD acts on another receptor, and that receptor has a signal transduction pathway that downregulates the CB1 receptor. So either it's a direct interaction of THC and CBD on the same receptor on CB1, or it's on two receptors and it's an interaction in the signal transduction pathway. Fascinating, actually, area of research.
All right, Audrey, it looks like we had several questions pop up in the chat box. Audrey or Jeff?
Yes, hi Kathy, Audrey again. So it looks like our next question came from Jim McCrae, and I think there's multiple parts to it. So maybe Jim, if you just want to, I saw you sent a second message, elaborate on it. If you want to unmute and explain your question, go ahead. Okay, I guess I'll read it out then. If folks are having trouble unmuting too, please put that in the chat because you should have the ability to unmute. But okay, so Jim writes, you mentioned the variation across individuals in their reported experience with cannabis. What of the well-known and very wide variation in effects across different strains of cannabis? Does the breadth of impact across 20+% delta-9 THC strains not suggest complex cross-cannabinoid impact on drug effects? And then a second part, to expand on where my question was targeted. While reductionism is convenient, it's not possible, is it not possible, excuse me, that the entourage effects often mentioned in describing the medicinality of cannabis are also at play in the expression of impairment in cannabis? Might not some cannabinoids, for example, be protective of delta-9 mediated impairment, excuse me. So I know that was multiple parts.
There's the famous word entourage effect, which was such a good word that was, but truly the definition of entourage effect is the definition of drug-drug interactions, is the fact that when you actually have two drugs, they're going to produce something different than only when you have one drug. So I mean, that's the only definition that I can come up. I think it's a great word, but we're just talking about drug-drug interaction, two drugs that are actually producing a different effect than a single drug. I would say that the tempering effect of CBD on the THC CB1 response is one of the most exciting areas of research right now that people are looking at. How can we, and we don't know if it's only because it will simply, as my previous explanation, the two molecules are acting on the same receptor and therefore it's simply tuning down, or if it's a more complicated, I would call it even therapeutic effect, where CBD is actually tempering the effect of the entire signaling pathway. And I think CBD is a great molecule, but I think the medicinal chemists that we have can probably optimize it. And we can have even more potent CBD molecules that might actually have even more promising therapeutic effects.
One thing to add to that too, the question also talked about other cannabinoids as well. And I mean, what Neffy just described is what's known for CBD. None of that is known for any of these other compounds that are cannabinoids. It's also true that the plant produces many other compounds. Terpenes, for example, are one that everybody's familiar, most people are familiar with. But there's other classes of compounds that it produces as well, flavonoids and some other things. We don't know what any of those do. And as you change the concentration of these compounds relative to each other, or as was discussed, very large range of THC levels, you throw in very large range of differences of all these other different compounds, and that drug-drug interaction that Neffy was just talking about, it's probably infinite possibilities of what could happen. It's going to be very difficult to tease that all apart, to be honest with you, when you're talking about when you're ingesting, especially a raw extract or especially the raw plant material. It's not one compound. It's hundreds or thousands of compounds that you're ingesting or inhaling, and that's a very complicated mix. Brad, you were going to say something too?
No, I think you covered what I was going to say, David. I was more going to recognize that Jim's question recognizes the layers of complexity that are at work here. And we're just peeling back some of the first layers of the onion. You can go so deep on this with polypharmacy and drug-drug interactions. And we're not even really talking about, is everybody at a similar level of impairment or lack of impairment as individuals? I mean, is everybody's baseline the same? We could go down that rabbit hole too. So all this complexity really makes it challenging to find a starting point for what is impairment, how do we define that functionally for policy purposes, and it's certainly a challenge.
Thanks for those questions, Jim. I also see a couple hands raised. Am I seeing that correctly, Audrey and Jeff?
Yeah, I'm seeing one hand raised right now. So
All right.
should we go ahead and call on Gabriel
Yeah.
instead of going? Okay, perfect.
Yeah,
Go
let's
ahead,
do that.
Gabriel.
Hello. Am I muted?
Hello.
We hear you.
Yeah, hello. My name is Gabriel. I work for the federal regulator in Canada, so at the Office of Cannabis Science and Surveillance. I'm at it's 5:30 here, so I'm in the middle of kid pickup. So I'll try my best to ask my question. And I apologize that I may have missed some parts of the conversation. So I'm happy that they're recorded, and I'll appreciate the link. But yeah, so this is a really fascinating topic, and this is also something that we're thinking about in Canada, particularly because we have a very strict limit on the THC limit, the quantity in edible cannabis. We obviously don't have this kind of same extent of an issue because of our definition of hemp, but it's definitely something that we're starting to see more and more in the regulated market. And I'm not a pharmacologist, so maybe this will sound a little naive for the academics on the call, but I'm just wondering, like, if this is something that would be possible. I've seen that in the field of toxicology, where one molecule that is more or less well understood can be kind of set as the baseline, and then other molecules are kind of compared to it, and you can derive an equivalency factor for it. I think I forget what molecule, but it might be digoxin or some other chemicals, and obviously I think those are simpler molecules than cannabinoids. But do you think that it would be possible, or do you think that there could be studies that could be like developed in a way that are just for that, you could compare other, whether it's delta 8 or delta 10, but you can compare other cannabinoids that act on the CB1 receptors, and you could kind of compare it back to delta 9 that's well understood. You can look at equivalency factors. So for regulators, when you're trying to, let's say that you're trying to keep the total, like the THC equivalent under 10 milligrams, then you can kind of easily do the math with, okay, it has 9 milligrams of delta 9, so we shouldn't allow more than another one equivalency. That might be a little naive, but as a regulator, that would be perfect. So yeah, maybe I'll leave it to you.
I might respond first. Gabriel, thank you for being here from north of the border. We certainly appreciate you being here, asking the question. And I think here in the States, we could learn a lot from what you and Health Canada have done in terms of how we're regulating cannabis. what I would suggest, and Neffy is probably a better person to answer this, but if we have a solid functional test, then we can have relative ratios or the sort of equivalency that you're describing, Gabriel. And another example there is sweeteners. All your artificial sweeteners have sucrose or sugar equivalencies. And I think if you do have a solid functional test, you can create that system of equivalences.
And that works if they function similarly. So if they get enough modifications to the molecules that they still have some of that effect, but then do something else as well, then it starts to get a little complicated. I know we don't really want to hear that, but it'd be nice if everything was simpler, but it's not. Anyway, Neffy, you want to add to that, or?
No, I think both of you did a good job on outlining it's a good idea to actually use a benchmark for bioactivity and then figure out equivalency. Something that would be twice as potent could be regulated in half of the amount. It's a really good idea. Once we have actually the entire equation, this molecule of THC does this behavioral impairment, based on this reference, we might actually have this approach. Very interesting idea. Thank you for sharing that, Jim.
All right.
Yeah, thank you. Thanks everyone for the answers.
Thanks very much. Audrey, I can see the chat, so I'm gonna go ahead and we're running out of time here, so I'm focusing in on questions that pertain to our discussion today, and that's about what's impairing and not impairing. If you have questions about LCB packaging and labeling, please reach out to that program. Because I see a collection of questions, some that have to do perhaps with other things beyond the conversation today. But here's a question from Annie, and we've only got about 4 or 5 more minutes here. If a cannabinoid is naturally occurring in cannabis, but not at high enough levels needed for products in the market, but it can be artificially synthesized from a different cannabinoid and is the exact same molecule as the naturally derived molecule, CBN, for example, how is that artificial? Requires it to be in a separate bucket and not allowed when it is the same molecule with the same effect or impairment? And I think that might be going into what's artificial and what's not.
Yeah, we've had conversations about that.
Yeah.
And I think that how I like to explain it to people is you have, let's say, grapes can be turned into wine, and so if you look at this, you can take a naturally occurring cannabis and turn it into something that is naturally occurring, and that synthesis, and whether or not that's allowed and is a longer term rulemaking. I don't think that's part of. And then you have things that, like a Jolly Rancher that's never seen a grape plant, and those are completely artificial cannabinoids. So you have, natural cannabinoids you can produce, natural cannabinoids through the synthesis as well, and then you have artificial that are just never really been natural occurring. I don't know if somebody else has a better.
Yeah, I've been using this actually differentiation between phytocannabinoids that are produced by the plant versus artificial cannabinoids which are not produced by the plant. However, we can synthesize phytocannabinoids in the lab, and we can synthesize artificial cannabinoids in the lab. So the word synthesis, synthetic cannabinoids, is not a really good word, because that just means that we're building it in the lab. I prefer to use phytocannabinoids produced by the plant and artificial cannabinoids that are not produced by the plant. So in this particular example, CBN, which if I remember well is actually a degradation product, is a phytocannabinoid, even though it's been degraded, and we can probably synthesize CBN, the phytocannabinoid, in the lab, but it's not an artificial cannabinoid because it exists in the plant.
Okay. Go ahead, Brad.
I was going to say, Andy, great question. It could serve as the basis for at least one more deliberative dialogue, probably 5 more. So, yeah, excellent question.
All right, so we truly only have 5 minutes left, and there's one other additional question here that I want to touch on, and then I need just about 2 minutes to close us out because I have some additional information I want to share. But with that in mind, Shannon Vito, VETTO, asks, can this panel define impairment for regulatory purposes at an individual cannabinoid level for the following list of CBD, CBG, Delta 8, Delta 9, and others? And I think that's what we're trying to do today, or at least start the combo.
Yeah, I think the answer to that, Shannon, is yes. Of the known cannabinoids, us on the panel could say yes impairing or no impairing. So you have Delta 8, Delta 9 as part of the question. Things like HHC, we talked about 11-hydroxy-THC, some other THC analogs like THCP and B. Probably put those all in the impairing bucket. CBD, CBG, CBN, non-impairing. I think that's relatively easy to do. We'll put that in air quotes. It's what we don't know, or the compounds we aren't aware of yet. That's the challenge. How do you define policy to incorporate those that may be discovered are produced by cannabis tomorrow, and could be produced by somebody in the laboratory and be placed into cannabis products? That's the real challenge.
And I think the impetus for all this is because of what happened with Delta 8, right? We knew it existed, but it wasn't studied very well. It wasn't being used because it's only produced at very, very low levels in the plant. But then somebody figured out how to make it in the lab from other compounds from the plant. And then all of a sudden it was very available. And how do we deal with the next one of those? That's the question that everybody's concerned about. When the next something like Delta 8 pops up, we want to be able to jump on it. And the big question here is everybody's concerned about our children, especially, right? We want to protect children and others. How do we make sure that we can do that? And how do we give the authority to those that need to regulate it, the authority so that they can?
Absolutely.
I agree.
All right, so on that note, we've got 3 minutes left. We'll go ahead and conclude the discussion for today. What I'd like to do is offer for questions that we might not have gotten to during this discussion today. If I can reach out to the panel with those questions, and perhaps we can develop some responses and add them with our recording of this session that we put on our website, if that's all right with the 4 of you. I just wanted to run that past you before I said we would do it. Okay. So,
Sure.
So there's that. If you have additional questions, please send them to rules. That's RULES at lcb.wa.gov. You can also find that on our rules website on our LCB outward facing website. The other thing I wanted to offer is some additional deliberative dialogue dates. I'm going to share my screen really quickly here. So we currently have 2 additional deliberative dialogue sessions scheduled, one for May 31st of 2022 and the next for June 21st of 2022. So I'm going to leave that up there for a second or two. To that end, we want to make sure that we're approaching this topic from a lot of different perspectives. So if anyone in the audience would like to suggest someone for a future panel, please don't hesitate to reach out to the rules staff. We're happy to take a look at your suggestions and do some reach outs to in-panel our next sessions. So again, you can send those suggestions to rules at lcb.wa.gov. I'm going to stop sharing. All right, so turning it back to the panel really quickly, we have 1 minute left. Anything else you'd like to share before we close?
No, I guess just thanks everyone for listening.
Thank you for the
Right.
questions.
That discussing, right? I think the deliberative dialogue format is important, that we have these discussions. It's important that you have them probably with people that you don't agree on them with. I think that's what I'd say. A lot of this stuff needs to be figured out, and it'll only be figured out through these sort of intelligent discussions and learning from each other.
Absolutely.
All right, thanks to y'all. Sir, you're on camera. If there's a question that you'd like to send us at rules, we'd be happy to answer it, but we need to close the session for today. So thanks very much for joining us. And with that, I will go ahead and we're going to go ahead and stop recording. Thanks everyone for your time today. You can look for this session again recorded on our outward facing website, and wishing everybody a great day. Take good care, everyone. Bye.